Hello students, I am Dr. Meenakshi Mahur, Assistant Professor, Zoology, Sri Savliaji Government College, Manfia, Chittagong. Today my topic is mutation. It is part 2 in which I will discuss numerical chromosome aberrations, and in this part I will discuss mainly aneuploidy. So it is just a revision. Mutation is of two types, intergenic and intragenic. That is, intergenic means chromosomal mutation and intragenic means gene mutation. Chromosomal mutation is of two types, structural chromosome aberrations and second is numerical chromosome aberrations. They are also known as genometric mutation. I have already discussed this part in the previous lecture. Now today I am going to discuss this part, numerical chromosome aberrations, which includes aneuploidy and euploidy. So what are genometric mutation? They are the mutation in which numerical changes in chromosomes or variation in chromosomal number occurs, and they are also known as genometric mutation or numerical chromosomal aberrations. The variation occurs in the chromosome number is due to error in meiosis and mitosis. I will discuss this point in later slides. First of all, what is ploidy? So it is the number of basic chromosomes set in a cell is known as ploidy. For example, a diploid cell, it has two sets of chromosome. That's why it is known as diploid. If a cell is with 6 sets of chromosomes, then the cell is known as hexaploid. So this is ploidy. Now, genometric or numerical mutation or numerical chromosome aberrations are of two types. It includes euploidy and aneuploidy. Euploidy consists maybe monoploidy, diploidy, polyploidy. And polyploidy again may be autopolyploidy, allopolyploidy or autoallopolyploidy. Euploidy I will discuss in next lecture. Today my focus is on aneuploidy. Aneuploidy may be hyperploidy or hypoploidy. Hyperploidy means they may be trisomic, double trisomic and tetrasomic. It may be monosomy, double monosomy or nullisomy. So what is aneuploidy? Aneuploidy means changes in one or more chromosome number in a set of chromosomes. Euploidy means changes in a whole chromosome sets. So aneuploidy is a condition in which one or more chromosome is added or deleted from basic chromosomal number in a diploid cell. Basic chromosomal number in diploid cell is 46 in human being. In aneuploidy, the chromosome number is not multiple of basic chromosomal number. The organism with such abnormal conditions with aneuploidy are known as aneuploids or heteroploids. So first I will discuss hyperploidy. Hyperploidy means addition of one or more chromosome to a diploid set. It may be trisomy, double trisomy and tetrasomy. So first is trisomy. It is 2n plus 1. 2n means 46 chromosome or two sets of chromosome. 2n means diploid. So 2n plus 1. If we talk about human being, then 2n means 46 chromosome or 23 pairs of chromosome or 46 number of chromosome. If we talk about trisomy, then 2n plus 1 means 46 plus 1, that is 47 chromosome in a cell. So trisomic are those organisms which contain one extra chromosome in addition to the diploid set of chromosomes. One extra chromosome in one pair of chromosomes. Example, Down syndrome. It is also known as trisomy 21. It is due to an extra chromosome number 21. It means there are 21st chromosome, and the individual with Down syndrome have 46 chromosomes. So they have a 46 plus 1, that is it's equal to 47 chromosome. So a person having Down syndrome will have 47 chromosome, 46 plus 1. Second is example is Klinefelter syndrome. It is again characterized by 2n plus 1. It means 46 plus 1, that is equal to 47. So here 44 the autosomes and 2 sex chromosome, so it becomes 46. Now one more chromosome, so there is an extra chromosome, that's why it has 47 chromosome. So its genotype is 44 plus XXY. How it occurs? It occurs when an abnormal egg, that is XX, fuse with a normal sperm Y. So it has extra chromosome X, means it should be there should be fusion between X and Y, but there is a fusion of XX and Y. So one more extra chromosome, that is X, is a extra chromosome in Klinefelter syndrome. So 44 is autosome plus X and Y, 46 chromosome, then one extra chromosome X, so it becomes 47. Then Edwards syndrome, it is a trisomy 18. In Edwards syndrome, again 46 plus 1, so 46 plus 1 extra chromosome at the 18th homologous pair. So there are 3 18th chromosomes. So it is Edwards syndrome. Then double trisomic, that is 2n plus 1 plus 1, means 46 plus 1 plus 1. If a cell contains one extra chromosome in two different pairs, that is one extra chromosome in two non-homologous pair of chromosomes. For example, if there is a extra chromosome at the 22nd position, so 22nd chromosome, so 3 22nd chromosome. If there is one more extra chromosome like at 15th chromosome, then there are 3 15th chromosome. So one extra chromosome in two non-homologous pair of chromosome, one extra chromosome in two non-homologous pair of chromosome, then it will be double trisomy. Means there are trisomy at two places, at two non-homologous pair of chromosomes. Now tetrasomic, that is 2n plus 2. If a cell with extra pair of chromosome, is the extra pair in this one extra chromosome in two non-homologous pair, but here cell with extra pair of chromosome, means if there are 23 pairs in human being, if there are 24 pairs of chromosome, then chromosome will be 48. If 46 plus 2, 47 and 48, so 48 chromosome. So a man with two extra chromosome, or a cell with extra pair of chromosome, it may be tetrasomy 9p, tetrasomy 18p, tetrasomy 12p, which is also known as Pallister-Killian syndrome, tetrasomy 22, Cat-Eye syndrome, extreme Klinefelter syndrome, where you can see 44 plus XXYY. So 44, 45, 46, 47, 48. So there are 48 chromosomes, but, or we can say 24 pairs of chromosome instead of 23 pairs of chromosome. So it is a tetrasomy. And in this case, there are 23 pairs of chromosome, but with two extra chromosome. in two non-homologous pair of chromosomes. So it is a hyperploidy because here you can see there is addition of one or more chromosome to a diploid set of chromosomes. Now hypoploidy, it is a deletion of one or more chromosome from a diploid set. So first is monosomy, that is 2n minus 1. If there is a loss of one copy of chromosome from a diploid complement set, then it is a monosomy. For example, Turner's syndrome. Here you can see 44 plus XY, X instead of 44 plus XY or XX. So there are 44 plus 1, only 45 chromosome instead of having 46 chromosome. So this is a 44 plus X. How it occurs? It occurs when an abnormal egg that without any gamete fuse with a normal sperm X, then it becomes XO plus 44 autosomes. So the individuals will have 45 chromosome, 44 autosome and only one sex chromosome, that is a 1X instead of pair of the whole pair of chromosomes. So it is a monosomy. Now double monosomics. When a cell misses two homologous chromosomes from two homologous pairs. When a cell misses two non-homologous chromosomes, then it becomes double monosomy. That is 2n minus 1 minus 1. Means one chromosome is missing from two non-homologous pair of chromosomes. So double monosomy mosaicism has been reported in a retarded child with multiple congenital malformations. So there are only 44 autosomes. Two chromosomes are missing from the whole set of chromosomes. That means one copy of chromosome, two from two non-homologous pair of chromosomes. Now 2n minus 2. There is a loss of one homologous pair of chromosome. It is 46 minus 2 is equal to 44 chromosomes. Means there are only 20. If we talk about human being, there should be a 23 pairs of chromosome, but in nullisomy there will be only 22 pair of chromosomes. It is a lethal in diploid. It can be tolerated in wheat, which is a hexaploid. So it is a hypoploidy. So in this example, in this monosomy, double monosomy, nullisomy, you can see there is a deletion of one or more chromosome from a diploid set. And in hyperploidy, there is addition of one or more chromosome to a diploid set. It can be trisomy, double trisomy and tetrasomy. Trisomy 2n plus 1 means 46 plus 1, that is 47. 2n plus 1 plus 1 means 46, 47 plus 1, 48. So 48 chromosome. 2n plus 2 means again 48 chromosomes, but extra pair of chromosome means there are 24 pairs of chromosomes. Here monosomy 46 minus 1 means individuals with 45 chromosomes. 2n minus 1 minus 1. Again a person or individual with 44 chromosome means 2n 46 minus 1 minus 1. So 44 chromosomes only. Nullisomy 2n minus 2, again a person with 44 chromosome instead of 46 chromosome. So in today's lecture I gave a brief idea of classification of mutation, then what is genomic mutation or numerical chromosomal aberrations, then it consists of euploidy, it may be euploidy or aneuploidy, and I discussed aneuploidy today. Then what is aneuploidy? That is a changes in a one or more chromosome number in a set. It's called aneuploidy. Then this is a chart showing hyperploidy and hypoploidy. Then what is the causes of aneuploidy. So, main cause of aneuploidy is non-disjunction. Actually, it is the condition in which one or more pair of chromosomes, that is bivalent chromosomes, fail to separate during anaphase of meiosis first. And because there is irregular distribution of chromosomes at poles, one daughter cell receives one or more extra chromosome, while other daughter cell lack one or more chromosome and they form respective gametes. Means one daughter cell with extra chromosome and one daughter cell, another daughter cell with lacking one or more chromosome. So when the gamete with extra chromosome fuse with a normal haploid gamete, then what will be result? It will result in hyperploids. While when the gamete lacking one or more chromosome fuse with the normal haploid gametes, then it will result in a hypoploid. Another causes of aneuploidy is non-orientation of one or more bivalent at metaphase first of meiosis first, or there may be loss of individual chromosome in meiosis or mitosis, or there may be irregularities in segregation of chromosome during meiosis in polyploidy condition, or multipolar mitosis with irregular distribution of chromosome to daughter cell. So here is a diagram which is showing non-disjunction. So if there is a normal X chromosome segregation, then first meiotic division occur. One chromosome here in one daughter cell, another chromosome in another daughter cell. After second meiotic division, four daughter cells are formed and each with one set of chromosome, or they are haploid. Each gametes carry one set of chromosome. Now, non-disjunction. If it occurs during meiosis first, then what will happen? You can see. So this is a diploid cell. Now, first in this diagram, you can see one daughter cell get chromosome, but another daughter cell is not getting any chromosome. So from this daughter cell, two daughter cells again are formed after second meiotic division. So after second meiotic division, you can see this gametes will be without any chromosome. But this gametes, in this gametes you can see there are the double chromosome. So instead of having X only, there is XX. In this again you can see XX instead of single X. It should be like this, but now you can see this is XX, XX and this is a 0, 0. If non-disjunction occurs during meiosis second, then what will happen? There is a proper distribution of chromosomes. So one daughter cell receive one and another receive also one. So now they are haploid, but after second meiotic division, you can see this is with double chromosome, this is without any chromosome, this is haploid getting one chromatid, and this is again one chromatid. So it is a normal, but there is a non-disjunction. So daughter cell, so this gamete will be with extra chromosome, and this gamete this gamete is lacking chromosome. So instead of this, you can see this one is a non-disjunction, means when the pair of chromosome fails to separate. It is also a diagram which is showing non-disjunction. Thank you.